This work is important because the technology that we developed doesn't just help the problem that we're interested in, the unfolding and refolding of proteins, but it's broadly applicable across a wide range of biomedical problems, as well as nanotechnology and nanoscience.
A lot of diseases are caused by when proteins misfold. And so understanding that process is really important.
We're at JILA, which is a joint institute between NIST and the University of Colorado.
This is our AFM laboratory. The kind of measurements we do are to study the folding and unfolding of proteins. We do this with atomic force microscopy, where we apply force to the molecules to watch how they fold and unfold. An AFM cantilever is this small strip of metal, and it has a long pointy tip at one end that allows us to push it very precisely with sort of extremely fine resolution onto a surface.
So it's very thin, it's very flexible, and one of the problems that people have had is that these cantilevers tend to drift over time. The cause actually was rather surprising. It's the metal coating. So by removing the metal coating that everyone thought was essential to being able to do high resolution measurements, we found that we made the stability of the microscope much better. A second innovation that we made was taking these cantilevers and not using the ones that industry gives us, but using what's called a focused ion beam to carve out the cantilevers, to make them smaller, to make them have less hydrodynamic drag.
So we find that by modifying commercially available cantilevers, we're able to improve how stable they are, how precise the measurements we can make are, and perhaps most interestingly, the time resolution with which we can make those measurements.
And collectively, these set of improvements have allowed us to study the unfolding of membrane proteins with 100 times higher time resolution and 10 times higher force precision. And as a result, we see all sorts of dynamics.
We're greatly expanding the information that we have about how proteins fold and unfold, and that may one day help us understand how to prevent them from misfolding. So misfolding of proteins are implicated in certain diseases such as Alzheimer's and mad cow disease.
It's this merger between doing the investment in technology and instrumentation and developing that, and then now sort of the real excitement happens as we get to do all this interesting science that was the promise of this technology, and it's what kept us motivated, but now we actually get to go do the really fun stuff.